Now, in the previous lecture, we studied about the role of renin in the management of hypotension. We studied that the renin, which is an enzyme which is being released from Purkinje cells, and it comes out from efferent arteriole and it will enter into the main circulation of the body. Now, for example, hypothetically, if this is the main circulation of the body, this will come out and it will enter into the main circulation. This renin will come out from efferent arteriole and through renal vein it will enter into the systemic circulation. Now, since this renin is an enzyme, it will act upon some substrate molecules, and the most important substrate molecule in this systemic circulation will be angiotensinogen. So, where from this angiotensinogen is released? That is actually released from liver. If this is liver, this has vast number of hepatocytes. Liver is made of vast number of cells. These are called hepatocytes, and these hepatocytes, they produce a lot of proteins, not all proteins, a lot of proteins, and out of them some proteins, these are called albumins, globulins, especially alpha, beta, but you should know that only alpha and beta, not gamma. Why? Because gamma is actually an antibody which is actually coming from the plasma cells. It releases an important substrate, that is, I can say, angiotensinogen. This angiotensinogen, it is not actually released at some instances, it is released continuously inside the body, So, if we are standing here, you are sitting there, angiotensinogen is being released from the liver cells continuously throughout the day and night. Now, what happens? This angiotensinogen, it is actually alpha-2 globulin. This is substrate for this enzyme renin. What happens? This renin, it will act upon this angiotensinogen. So, if I write this process here, I will say that angiotensinogen, it is actually inactive form, inactive substrate that is being converted into angiotensin 1, or I can write it here as angiotensin 1 or angiotensin 1, which is active form that is being formed by this renin enzyme. Now, what happens here? This angiotensin, so I will say this angiotensin 1 is formed in the systemic circulation. Now, this angiotensin 1, it has to move towards the lungs, where from it is being converted into second form, that is called angiotensin 2. So, I will say that it will move through the lungs, and from lungs it will be converted into angiotensin 2. This is this angiotensin 1, that is decapeptide. It means that peptide, that polypeptide which is having 10 peptide chain, and this is octapeptide. How this happens? Now, I will say this is actually, if this is lung, I will show it from one side. What happens? This lung, inside there are various bronchioles. These are the bronchioles present in the lung, and you know that every bronchiole is having alveoli at the tip. And from there are capillary network outside the alveoli. For example, if I take one alveolus here, I will say that this alveolus is like this, and outside that there are various capillary networks outside from that area. As soon as this blood it moves through this lung, if I enlarge this diagram, I will say that the endothelial cells are these capillaries inside the lungs. These are having some specific cell types, endothelial part inside the lungs. So I will say that this angiotensin 1 will move toward the lungs, where inside the lungs these capillary endothelium are having endothelial cells, special type of cells, and these special type of cells have on the outer surface of the cells an expression of enzyme, and that expression of enzyme is actually called AASE. So AASE is not actually present inside the alveoli or bronchioles or bronchi, no. These are actually present in the endothelial, on the external surface of endothelial cells of capillary network or bed. As soon as this AT1 is moving through these, it is acted upon by these ACE molecules and it gets converted into AT2, that is angiotensin 2. This is decapeptide and this is octapeptide. So I will say from here, renin is responsible to convert angiotensinogen, that is inactive, into active form, that is AT1, and this AT1 when moves through the lungs, it is acted upon by the ACE molecules, that is angiotensin converting enzyme. These molecules convert AT1 into AT2. Now role of AT2 starts. Every function regarding this RAAS is performed by AT2. This is also active form, but AT1 is more potent than AT1. This is decapeptide and this is octapeptide. Now what happens? This AT1 is converted into AT2. This AT2 molecule, that is angiotensin 2, it will now perform three basic functions. Now if I say here, it performs three basic functions. One function, that is, it will act upon the adrenal gland. Second, it will act upon the angiotensin receptors. Third, it will act upon CNS part. So this angiotensin 2, which is formed, it will perform three basic functions. One, that is, it will act upon adrenal gland, which will be studied later. Second, it will act upon angiotensin receptors, receptors, and it will act upon CNS. All the three pathways contribute to the management of hypotension or management of hypertension. Now